Hello students, today we will study about the next or last plant of the Bryophyta, that is the Funaria. The systematic position of the Funaria, it belongs from the division Bryophyta, class Bryopsida, order Funariales, family Funariaceae, genus Funaria. Occurrence: the genus Funaria is cosmopolitan and occurs both in tropical and temperate regions of the world. It includes about 117 species, out of which about 15 species reported from India. Funaria plants are commonly known as cord mosses. It grows on moist sandy places such as damp soil, moist rocks, sandy banks, and sometimes on tree trunks and on moist walls. The most common species of Funaria is Funaria hygrometrica, which grows commonly in the hills in closed tufts on moist grounds, walls, rocks, and crevices. The most common site of occurrence is recently burnt soil. It has been reported that burning increases the availability of soil nutrients, which favors profuse growth of the Funaria. Next, that is the gametophytic plant body, means adult gametophyte. The gametophytic phase of life cycle of this plant includes two stages. First stage, that is the protonymal stage, formed by the germination of spores. Second stage, the adult gametophyte, developed on protonyma. The main plant body is the main plant body of this plant is adult gametophyte, which consisting of erect leafy shoot, that is this. It is commonly called gametophore. Vegetative structure. In the vegetative structures, first we study the external features. The gametophytic plant body is leafy shoot, which is short, about 1 to 3 centimeter in height. About 1 to 3 centimeter in height, and grows in tufts. The plant body of Funaria are differentiated into an erect axis, that is the stem, and spirally arranged leaves with numerous rhizoids. These are the rhizoids which are attached at the base of the axis. Rhizoids, that is this or this. The rhizoids arise from the protonyma, that is the first stage of the gametophytic of life cycle, as well as from the base of adult gametophyte, that is this. The rhizoid originates as a tubular elongation. The rhizoids originates as a tubular elongation of surface cells at the base of axis. At the base of axis, it divides, that is the division, and redivides to form a long cylinder multicellular branched structure, that is this. It consists of branching rows of cells with oblique septa. With oblique septa. Students, you see here, that is the oblique septa. They usually contain oil and develop chlorophyll if exposed to light. Young rhizoids are colorless, but at maturity they develop brown color. The rhizoids enter vertically downward, that is this, vertically downward into the soil and form a tangled mass. The rhizoids fix the plant body to the substratum and help to absorb the water and minerals. The axis or the stem, that is this, that is this. The main stem or main axis is erect, cylinder and branched. The branches are extra axillary. Listen, the branches are extra axillary and monopodial. The branches usually arise from the basal portion, that is this, you see here. The branches usually arise from the basal portion of the axis and become vertically erect similar to main axis. Next, that is the leaves, that is this. The leaves are arranged spirally, you see here, that is leaves are arranged spirally on the main axis and branches. The lower leaves are small, that is this, the lower leaves are small and scattered, where the upper leaves are large and densely crowded at the apex. You see here, densely crowded at the apex. Each leaf is flat, that is this, each leaf is flat, bright green in color, ovate in shape, with pointed apex, that is pointed apex, and entire margin. The leaves are sessile and have well defined midrib Next, that is the internal structures. First, we study the internal structure of the main axis or the stem. The axis has simple internal organization of cells. The outermost layer is epidermis, which is one cell thick and consists of tangential elongated thick-walled cells and consists of tangential elongated thick-walled cells. These cells contain chloroplasts These cells contain chloroplasts. The stomata are absent. Epidermis is followed by multi-layered cortex. The multi-layered cortex. Cortex is consisting of parenchymatous cells. Outer cells of the cortex toward the epidermis in young axis contain chloroplasts, means perform the function of photosynthesis. These cells become thick-walled and lack chloroplasts as the axis matures. The center of the cortex, center of the main axis, is occupied by vertically elongated cells. These cells are slightly thick-walled, compact, haline, and often lack protoplasm. Means dead, becomes dead. This zone, that is this zone, serves the function of conduction and therefore called conducting strand. Next, go to the leaf. The internal organization of leaf is simple. It has several cell thick midrib and single layered wing and single layered wings. The center of midrib, that is this, the center of midrib is occupied by conducting strand made up of thick-walled cells. The midrib has upper, that is this, has upper and lower epidermis consisting of thin-walled cells that contain chloroplasts. The cells of the wings are polygonal and contain numerous large chloroplasts. Next, go to the reproduction. The reproduction in Funaria plants occurs both by vegetative as well as sexual methods. In case of vegetative reproductions, the vegetative reproductions occurs in primary protonema. Students, you see here, that is structure of primary protonema by the formation of colorless intercalary cells. कि प्राइमरी प्रोटोनिमा में फॉर्मेशन ऑफ कलरलेस इंटरकैलरी सेल्स, और जो ये कलरलेस इंटरकैलरी सेल्स है दे आर नोन एज द सेपरेशन सेल्स. कुछ टाइम के बाद दी सेपरेशन सेल्स बिकम्स डिजनरेट्स, जिसकी वजह से जो प्राइमरी प्रोटोनिम है broken into pieces and each piece develop a new plant. Next, that is the secondary protonema. The secondary protonema may be formed from any cells of detached, listen here, the secondary protonema may be formed from any cell of detached stem from any cell of detached stem, leaf, and rhizoids under moist conditions. These filamentous protonema may develop buds, and these buds serve as the means of vegetative reproductions. That is the next way of vegetative reproduction, that is gamete. The terminal cell of filamentous protonema, the terminal cell of filamentous, ये प्रोटोनिमा है, ये फिलामेंटियस है. The terminal cell of filamentous protonema may develop small bud-like structures called the gamete. और जो ये गैमे है consist of thin walled cells filled with chloroplasts. जैसे ही ये गैमे डिटैच होंगे फ्रॉम द फिलामेंट्स एंड डेवलप इनटू ए न्यू प्लांट. नेक्स्ट, that is the gamete-like structures have also been reported to occur in subterranean rhizoids. कि subterranean rhizoids which are present underground in the soil, कि उन underground subterranean means subterranean rhizoid से gamete-like structures कभी कभी प्रोड्यूस हो जाते हैं, ऐसा कुछ रिपोर्ट किया गया है. एंड दीज आर टर्म्ड एज द ट्यूबर्स, एंड दीज ट्यूबर्स बिहेव लाइक गैमे एंड ग्रो इनटू न्यू प्लांट्स. नेक्स्ट वे ऑफ वेजिटेटिव प्रोडक्शन, दैट इज एपोस्पोरी, मीन्स एपोस्पोरी वे से, कि सेकेंडरी प्रोटोनिमा may also arise from any cell of sporogonium. Means the secondary protonema may also arise from any cell of sporogonium. They are similar to those produced from gametophyte. कि वो जो उनका स्ट्रक्चर है, मॉर्फोलॉजी है, they are similar to those produced from gametophyte and developed into leafy gametophores. इस टाइप का जो प्रोडक्शन ऑफ प्रोटोनिमा डायरेक्टली फ्रॉम स्पोरोगोनियम सेल्स इज टर्म्ड द एपोस्पोरी. The gametophores produced from such protonema are diploid in nature, but resembles in form and structure with haploid gametophores. Next, that is the sexual reproduction. The plants are monoecious, autoecious. Monoecious and autoecious means the antheridia and archegonia are borne on separate branches of the same plant, and protandrous. Monoecious, autoecious and protandrous. Protandrous का means the antheridia develop first and archegonia later. Protandrous means the antheridia develop first and archegonia develop later. The sex organs are borne in groups. The sex organs are borne in groups at the apexes of branches. The sex organs usually mature in autumn or winter. The formation of sex organs is induced under certain critical low temperature. The formation of sex organ is induced under certain critical low temperature, जबकि the specific photo periods have no effects. जबकि specific photo periods have no effects, mean duration of light have no effects. Next, that is the antheridia. The antheridia are born in groups. The antheridia are born in groups at the apexes at the apex of main axis, which functions as the male branch. The apex of branch is somewhat convex and bears a cluster of antheridia. The cluster is surrounded by large perigonial leaves, which are roasted in the form of a shallow cup, roasty shape में, in the form of shallow cup-like structure, that is this. The center of cup is reddish in color. The antheridia are intermixed. The antheridia are intermixed with a large number of sterile paraphyses. Each paraphysis is unbranched, multicellular, means 4 to 5 cells, filamentous, hair-like structures. The terminal cell of the paraphysis is large, nearly spherical or capitate, and capitate. The other cells, जो उसकी अदर सेल है पैराफाइसिस की टर्मिनल सेल के अलावा, are narrow, elongated, green, chlorophyllous and photosynthetic. The paraphysis protect, जो फंक्शन है इसका, द पैराफाइ, एक तो इसका फोटोसिंथेसिस करना फंक्शन हो गया ड्यू टू द प्रेजेंस ऑफ द क्लोरोफिल. Second, the paraphysis protect the developing antheridia. They retain water in between. Next function, they retain water in between and secrete mucilage, which helps in dehiscence of mature antheridium. The apical cell of the male branch, the reproductive branch, is used up in the formation of sex organs, either the antheridia or archigonia. Therefore, the further growth of the male or female shoot stops. Next, that is the structure of mature antheridium, that is this. The mature antheridium is differentiated into a short stalk, that is this, and a long club-shaped body and a long club-shaped body. The body is reddish brown or orange in color. It has a single layered it has a single layered style jacket which encloses a mass of androcytes. Here you see the androcyte mother cells, and the androcyte mother cells produce the androcytes. The apex of antheridium has one or two celled, that is cap, one or two celled, colorless, thick walled cells of cap, जिसको cap कहा, और दूसरा नाम that is the operculum. Each androcyte differentiates into biflagellate. Each androcyte differentiates into biflagellate anthrozoid. Next, that is the dehiscence of antheridium. The dehiscence of antheridium occurs by the help or the presence in the presence of the water. The water collects in the perigonial cup. Water collects in the perigonial cup, and the water collect in the perigonial cup provided by the dew or the rain. कि वो वाटर आता कहाँ से है, चाहे तो वो रेन से आए या ड्यू से आए. द ओपरकुलर सेल और द कैप सेल, कि जो ओपरकुलर और कैप सेल्स है, एब्जॉर्ब द वाटर, and after the absorption the cap cell become mucilaginous and swell. Finally they are broken and form a small narrow pore at the distal end of antheridium. The mass of anthrozoids, that is this, still enclosed in the vesicles, VESICLE, oozes out through this opening or through this narrow pore. Gradually, the entire content comes out and the antheridium becomes empty. कि इसका जो कंटेंट है एंटायर, जो एंथ्रोजोइड्स का जो मास है, विच इज ओजीज आउट, बाहर निकल जाता है थ्रू द नैरो पोर, जिसकी वजह से जो एंथ्रीडियम है वो एम्प्टी हो जाता है. द एंथ्रोजोइड्स आर सेपरेटेड फ्रॉम द मास ड्यू टू द प्रेजेंस ऑफ लिपिड्स in the matrix and spread over the surface of water in the perigonial cup. जो perigonial cup है उसके अंदर उस कप में स्प्रेड हो जाता है, उसमें पानी हुआ वाटर है और वाटर के ऊपर वो स्प्रेड की फॉर्म में प्रेजेंट होता है, स्प्रेड हो जाता है. मतलब स्टूडेंट्स आज इतना ही कराएंगे. इन द नेक्स्ट वीडियो we will study about the archegonium and sporogonium. Thank you.